Assignment, I'll be designing and validating an expression vector. My gene of interest is the insulin gene, and the insulin gene codes for the peptide hormone insulin. The synthesis of insulin takes place in the beta cells of the pancreas. Insulin is secreted for the regulation of glucose levels in the plasma and has since been used for the treatment of diabetes mellitus. The aim of this project is to express the insulin protein in a prokaryotic bacterial cell. My gene of interest is the insulin gene, and my choice of vector is the PET blue one vector. I chose the vector because it has a strong promoter. It has an F1 origin of replication, which can create a single stranded DNA. The vector also has a laxygen, and it also has antibiotic resistance gene. Now for the insertion of the gene into the vector. Now the vector has already been digested using the restriction enzyme EcoRV. Now this EcoRV enzyme is used on the DNA molecule. So the restriction enzyme also acts on the DNA molecule, and it cuts the DNA molecule at the recognition site. And then T4 DNA ligase, the enzyme, is employed to join the vector with the gene and left to incubate for 1 hour at room temperature. Now, after incubation, the result is a recombinant plasmid. So the recombinant plasmid is the result of the ligation of the digested plasmid and the DNA molecule. Now, to transform the bacterial cells, I'll be using the heat shock method. The plasmid vector is supplied in the E. coli strain Nova Blue to maintain the integrity of the plasmid. The recombinant plasmid, that is the recombinant plasmid gotten from the ligation reaction, is transferred to the competent cells. Now for this project, I'll be using the E. coli BL21 DE3 PLACL competent cells. Now these cells are heat shocked in a water bath for 30 seconds at 42 degrees Celsius. An agar media consisting of ampicillin, IPTG, and Xgal is prepared, and the transformation mix is spread on the agar plate. This is left to incubate for 15 to 18 hours. The presence of white and blue colonies on the agar plate or on the agar medium is used to detect for the presence of transformed bacterial cells which took up vectors without inserts. The presence of white and blue colonies on the plate is used to detect for the presence of transformed bacterial cells which took up vectors without inserts. Now, because of the presence of the ampicillin resistant gene on the vector, it's possible that cells that have vectors without inserts will still grow. So this white and blue colony system is used to detect for the presence of transformed bacterial cells which took up vectors without inserts. Colonies containing vectors without inserts remain blue after incubation, and the colonies with vectors containing inserts remain white. Sanger sequencing is used to check if the correct vector is used. Sanger protein sequencing is used to check if the correct vector has been used for the project. Thank you.